Hello everyone. Today we are going to study about glycogenesis. It comes under glycogen metabolism in carbohydrate metabolism. So what do you mean by glycogenesis? Definition: it is a process of formation of glycogen or synthesis of glycogen. And this glycogenesis takes place in the cytoplasm of almost all cells, primarily liver and muscles. The requirements for glycogenesis are alpha D glucose, glycogenin or glycogen primer, energy in the form of ATP and uridine triphosphate and different enzymes. So now we are going to study the process of glycogen synthesis. In other words, glycogenesis. For the formation of glycogen, we require pre-existing glycogen primer. So there will be always a small fragment of glycogen available in the muscle and liver and other tissues. If there is no pre-existing glycogen molecule, then there is a protein which has enzymatic activity as well called glycogenin, which produces this small fragment of glycogen which is called glycogen primer. It has got alpha 1,4 glycosidic linkage, all these are, and there is a branch and the branching point is called alpha 1,6 glycosidic bond or linkage. So with this pre-existing glycogen primer, there will be a process of formation of glycogen which is called glycogenesis. So we need glucose, and this glucose phosphorylated to glucose 6-phosphate with the help of energy. So there will be hydrolysis of ATP to ADP with the help of enzyme glucokinase or hexokinase. So once glucose is phosphorylated to glucose 6-phosphate, this glucose 6-phosphate isomerized to glucose 1-phosphate with the help of enzyme phosphoglucomutase. Now this glucose 1-phosphate converted to what is known as uridine diphosphate glucose or UDP glucose. So we need uridine triphosphate which will be converted to UDP glucose. That means UDP will be added to glucose 1-phosphate. In turn, glucose 1-phosphate will be converted to UDP glucose. Simultaneously, it will release pyrophosphate, which will be eventually hydrolyzed to 2-inorganic phosphate. The enzyme for this particular reaction is known as UDP glucose pyrophosphatase. Now we have glucose in the form of UDP glucose. Now with the help of enzyme glycogen synthase, the glucose from the UDP glucose will be added to non-reducing end of this glycogen dimer. So here and here. You can see it will establish alpha 1,4 glycosidic bond. So what happens to this UDP? This uridine diphosphate will be released, which eventually can be converted to UTP, uridine triphosphate, with the hydrolysis of ATP. So this process continues. You can see glucose from ADP glucose will be added to this glucose end or non-reducing end one by one. So it will go on add glucose until there will be 10 to 11 glucose residues on one side. Again there is here non-reducing end. So it can go on add until 10 to 11 glucose residues are on either side. Okay, now this is nothing but elongated glycogen primer or pre-existing glycogen primer. Remember, this glycogen primer can be synthesized from glycogen, or there will be small fragments of glycogen in liver and muscle and in almost all tissues. So remember, glycogen synthase can establish only alpha 1,4 glycosidic linkage. So how many glucose it will be added? So there should be at least 10 to 11 glucose should be there on all the non-reducing ends of glycogen primer. This glycogen synthase cannot add branching point, that means alpha 1,6 glycosidic linkage. So what is the next step? Now we have elongated glycogen primer. So on either side of non-reducing end, there are 10 to 11 glucose are present now. Now there is another enzyme called branching enzyme. So this branching enzyme, it removes about 6 to 8 glucose. Here you can see 1, 2, 3, 4, 5, 6, 7. Around 6 to 8 glucose molecules are removed from one of the branching site. That means it breaks alpha 1,4 glycosidic linkage and establishes a new branch. So this point is called now alpha 1,6 glycosidic linkage. Okay. Between this glucose and this glucose is alpha 1,4. Between this glucose and this glucose is alpha 1,6. So now we have established one branch. Now again, glycogen synthase continues to add glucose from this non-reducing end until in this branch about 10 to 11 glucose residues, and also it will add here also until 10 to 11 glucose, and this end also there will be continuously glucose will be added from UDP glucose. So this process done by glycogen synthase, whereas branching enzyme, it removes 6 to 8 glucose molecules from the branch and establishes a new branch. So with the help of combined effort from glycogen synthase and branching enzyme, glycogen will be synthesized. Please remember the branch, the distance between one branch to another branch, that will be around 8 glucose. 7 to 8 glucose, there will be branch. Glycogen molecules are highly branched. Even it can establish branch here after 8 glucose, there can be another branch by branching enzyme. Compared to starch, there will be branch for 20 to 25 glucose, but in glycogen it is highly branched. Every 8 glucose you can see branch. So this is a simple explanation for glycogenesis. Please remember the rate limiting enzyme or the important enzyme for glycogen synthesis is glycogen synthase. No doubt it requires branching enzyme, but the rate limiting enzyme is glycogen synthase. Thank you very much.